Hi dear students, today we are going to discuss blue-white screening. Okay, blue-white screening. And the next method is a blue-white screening method. Okay, blue-white screening method. This method is widely used for screening of to get recombinant clones. Okay, this method is widely used for screening of recombinant clones. Okay, in this gene cloning procedure, there is a one vector is there. That vector containing a selectable marker gene, laxid gene, and an antibiotic resistant gene, ampicillin resistant gene. Okay, this gene cloning method use a vector called that vector containing a selectable marker, a laxid gene, and an antibiotic resistant gene, ampicillin resistant gene. Okay, that laxid gene encodes an enzyme called beta galactosylase. Okay, that laxid gene encode a protein called an enzyme called beta galactosylase. And that beta galactosylase cleave the substrate S-gal in the S-gal present in the medium to blue colored insoluble product. Okay, when we transfer this laxid gene into the bacteria, that time that bacteria will produce blue colored colony. Okay, that bacteria will produce blue colored colony due to the presence of this laxid gene. Okay, this laxid gene coding enzyme beta galactosylase cleave the colorless substrate S-gal which is present in the medium to blue colored insoluble product. Okay, this beta galactosylase cleave the substrate S-gal present in the medium to the blue colored insoluble product. Okay, when we insert a foreign DNA into the laxid gene, okay, when we insert a foreign DNA into the laxid gene, that laxid gene will be inactivated. Okay, that laxid gene will be inactivated. So that process is known as a insertion inactivation. Okay, when we insert a foreign DNA into the laxid gene, that time that laxid gene will be inactivated. So that inactivation process is known as a insertion inactivation. Okay, once this laxid gene is inactivated, that time this laxid gene containing colony cannot able to produce blue colored colony. Okay, once this inactivated laxid gene present in the bacteria that cannot able to produce blue colored colony. Okay, this laxid inactivated laxid gene only produce white colored colony. Okay, insert foreign DNA into this, okay, that laxid gene will be inactivated. Here you can see this is the a vector. Okay, this is the vector that containing the laxid gene here, and here ampicillin resistant gene. Okay, when we this is a foreign DNA, we'll insert the foreign DNA into the laxid gene. Okay, we'll insert the vector laxid gene will produce, we'll insert the laxid gene into the laxid gene of the vector. Okay, then after inserting this this foreign DNA into the laxid gene, we'll transfer this recombinant vector into the host cell. Okay, recombinant vector into the host cell, and so that time there's chances that we'll get some colonies. Here you can see this foreign DNA is correctly inserted into the laxid gene of this vector. Okay, this foreign DNA is correctly inserted in the foreign laxid gene of this vector. So this bacteria will produce, this laxid gene is absent, so inactivated. So laxid gene is inactivated. That means this is known as insertion inactivation. Okay, this laxid gene is inactivated, so this process is known as a insertion inactivation. And this bacteria laxid gene inactivated, that's why it's produced only white colored colony. Okay, only white colored colony, because this bacteria can able to survive, and this medium, this all medium will be ampicillin containing medium. Okay, ampicillin containing medium. Okay, this bacteria will be, this medium should be ampicillin containing medium, and this the vector containing foreign DNA in the lax gene that will produce blue colored colony in the ampicillin containing medium. Okay, why? Because it can able to survive in the ampicillin medium because it having a sequence a resin gene of ampicillin is here, so it can produce white colored colony. Okay, in this another one type of bacteria will also is with in this mixture, that's a having no insert in the laxagine. Okay, no insert in the laxagine, so that no insert in the laxagine, that's why laxagine will be active. Okay, laxagine will be active. So this bacteria this bacteria having what, this bacteria having the vector, but the foreign DNA is not inserted in laxagine, so that's why this bacteria laxagine is activated, that's why it's produce blue colored colony in the ampicillin medium. Okay, that's why it's produce a blue colored colony in the ampicillin medium. Okay, sometime this foreign DNA will be inserted outside this or inserted the outside the laxagine, that time also it will produce blue colored colony. Okay, sometime in this mixture some one bacteria also is there, that bacteria does not contain the vector, that type, that bacteria cannot able to survive in the ampicillin containing medium. Okay, that's bacteria cannot survive in the ampicillin resistant medium. Okay, so here you can see this is a blue colored colony. This blue colored colony is because of this foreign DNA is not inserted in the laxagine. Okay, the foreign DNA is not inserted in the laxagine. If foreign DNA is inserted in the laxagine, that laxagine will be inactivated. So the inactivated laxagine, that means that contain the foreign DNA, so that produce the colorless white white colored colony. So this white is the recombinant clone. So, okay, this white is a recombinant clone because there's a laxagine inactivated due to the presence or due to the insertion of foreign DNA to it. Okay, so we can select this white colored colony as a recombinant clone, and this blue is because there is no insertion of foreign DNA in the laxagine. Okay, this is the blue white screening method. Okay, this is the blue white screening method. Okay, from this, on the basis of the basis of color, okay, the basis of color, we can identify the recombinant clone in this blue white screen. That means the white color colony means there is a recombinant clone, and the blue means there is a unrecombinant clone. That means blue does not contain the recombinant DNA, and white contain the recombinant DNA. Okay, this is the blue white screening. So thank you for listening my class. Thank you.